Good morning and welcome. My name is Mike Fahey. I'm one of the managing directors here at the Wisconsin Foundation Alumni Association. And we're excited to welcome you to our program on the future of gene therapy. There is a lot of exciting work happening at UW-Madison in this area on gene therapy and using CRISPR, a relatively new tool in this scientific area. And we are thrilled to have one of our top researchers joining us today to talk about this program and gene therapy and what's happening here on campus. And not only are we going to learn a little bit of the science, but you'll also get a chance to visit his lab. We're going to go behind the scenes for a short view of how that work happens and how it gets done within the lab. And so it's a great opportunity, and we thank you for being part of this program. We welcome, as always, your questions and your feedback. So if you're joining us on YouTube, feel free at any time to drop a question into the chat, and we will leave as much time as we can at the end of the program to answer those questions and dig into the research and the work and the presentation. We're also being recorded, so if you want to go back and listen to the program in the future, you can do that as well. So I'm excited to welcome our presenter today, Dr. Krishanu Saha. Krishanu Saha is an associate professor of biomedical engineering and medical history and bioethics here at UW-Madison. He is also the McPherson Eye Research Institute's Retina Research Foundation, Kathryn and Latimer Murfee Chair. His lab is at the Wisconsin Institute for Discovery on campus, and he also participates in the Stem Cell and Regenerative Medicine Center and the Robert F. Holt Center on Science Technology Studies. Prior to coming to UW-Madison, Dr. Saha studied chemical engineering and biotechnology at Cornell University, University of Cambridge, and the University of California, Berkeley. So we're thrilled to have him. Thank you, Professor Saha, for joining us. And I think we're going to start by talking a little about gene therapy and CRISPR, and what is this CRISPR that we've learned a lot about?
Sure. First of all, thanks for the nice introduction and the opportunity to tell you about really the exciting work we have here on campus. CRISPR is really a popular term now, and it's related to these gene and cell therapies that you may have seen out there in the clinic. So there are approved gene therapies out here that have treated some inherited disorders on the left, like Luxturna and Zolgensma. On the right, there's T-cell therapies that have been genetically modified to fight cancer, and leukemias in particular. And the technology here is really technology that was developed, I would say, decades ago, arguably in the 1990s, to use viruses to put in genes into these either tissues or cells. And CRISPR is a new way of adding or modifying the genetic code. And just to start really as a foundational slide here, the DNA is in your body is 3 billion letters long, and a single misspelling can have profound consequences. So in this case, this is a T highlighted in red, and this is coming from a patient that has lost the ability to have central vision. So when the patient looks out and tries to focus on something in the center, they have trouble. This disease is called Best disease, or Best vitelliform macular dystrophy. And genome editing is the concept here is to fix bases in the DNA code. So in this case, you would move the T to a C, which is provides a healthy coding region for the protein that would be produced. And then in theory, this would rescue this person's vision if you were able to modify the genome in the right cells in the right part of the body, namely in the retina. CRISPR is a way of doing this, and this is a short cartoon that talks about the components of CRISPR. There's this orange molecule that you see here called the guide RNA, and a protein that's larger in purple on the left that's called Cas9 nuclease. On the right is a DNA template that flies out of the screen, but we'll come back later. The RNA in orange and the protein come together. They have affinity for one another, and if they're inside the nucleus of a cell, they scan the genome that's in teal here. And if it finds one of these mutant bases, that matches to the RNA sequence in orange. The protein has some enzymatic activity to cut the genome. And so you'll see the genome break apart, both strands of it. And once that happens, the cell takes over. It has in natural processes to detect DNA double-strand breaks. And if there's a repair template around, it can use that repair template to write in a specific sequence into that cut portion of the genome. And you see the repair proteins that are inside every single one of the cells that we have come in and repair that process based on the sequence in the template. And there's techniques here to do this not only for DNA, but also for RNA, mitochondrial DNA, and other molecules inside the cell. So this is really an abstract cartoon theoretical version of what happens. I wanted to give you a sense of what happens inside my lab. And so I was fortunate to have the staff at the foundation come follow me as I gave them a tour and record the beautiful space here at the Discovery Building. And what you'll see here is a really nice open space where we're sharing common instruments, materials, and freezers, et cetera, with a number of other labs. There's namely Sarah Gong, who's a nanomedicine specialist, Randy Ashton, who's a neural tissue engineer, and Tom Turing, who's a polymer scaffold expert. And the idea behind WIT is really to have collisions between these labs to spark new ideas. And really that indeed has been the case, especially around CRISPR, and I'll tell you two stories about that. What Ross here, our lab manager, is pointing to is some instruments that help us amplify templates that we saw in the cartoon. There's a polymerase chain reaction that involves heating and cooling of enzymes with DNA such that we can amplify a number of templates. Those machines over there can make on the order of 300 different templates at sufficient quantities such that we can use them for genome editing. And here now, Namita, a PhD student in biomedical engineering, is going to show you one of her cells under a microscope that has been genome edited. And so what she's pointing to are a number of small clusters of cells where genome editing has occurred. And she's moving around the bottom of the cell culture dish to point out particular colonies that have different growth characteristics. And so the goal of this genome editing was to rewrite portions of the genome that would confer a different way in which these stem cells would grow under these growth conditions. So you can see that she's moving around the plate, searching from well to well to find promising colonies that have been potentially edited. And this takes years of skill, and Namita's been doing this for several years now. So she has really expert eyes and a great sense of which cells to pick out. And what you see here as well is Namita changing the media or the liquid part that's covering the cells in cell culture. This is occurring in a biosafety cabinet because we want to make sure that no contaminating bacteria or viruses or pathogens are also growing along with our human cells. And so there's these safety measures that we take. These cells have a limited lifetime in these incubators if you don't change the media. The media has growth factors, proteins, nutrients, vitamins to help these cells grow. And every so often they use them all up, and therefore Namita and others in my lab, Ross, is preparing the media to put in fresh nutrients, growth factors, et cetera, to let these cells grow. And so this is a fairly routine process for many folks in the life sciences, growing cells outside the body, manipulating them, putting in genome editing characteristics. One of the key tools that we have is to add a drug in the media such that it only allows the cells that have been edited to grow out. So this type of drug selection is actually what Namita is looking for in this experiment. We've used CRISPR to put in a stretch of DNA that allows cells to grow in the presence of this drug. And it's really important. It's actually a key way in which we're able to have high quality control over the cells that we manipulate outside the body, such that we know exactly the genome and the sequence that would be inside these cells once we formulate a potential cell therapy that would go into a patient. So now that you've had a nice sense of kind of what CRISPR and this type of genome editing work is in practice here on campus, I think I'll delve into two stories where we use these types of CRISPRed cells to attack cancer. That will be our second story. And the first story is actually directly injecting CRISPR into a tissue. And we've done work where we've taken the RNA and protein that was in that cartoon, encapsulated them into nanoparticles, and then injected them into different tissues, namely the eye, the muscle, the liver, and the brain. I'll tell you one story about injecting into the eye. And then the second story will be how do we generate T cells that go after cancer inside the body. And the T cells have been essentially programmed using CRISPR to find and destroy cancer cells inside tissues within living patients. Both of these lean upon some national efforts that are shown on the right that I'll go into as well. So the first national effort is funded by the National Institutes for Health, and it is called the Somatic Cell Genome Editing Program or Consortium. You can see it spans both coasts, and Wisconsin has multiple projects within this center. We were actually the only center at the first phase of this that had multiple projects, so I think we're at the leading edge. And one of the beauties of working in such a team and science type of environment is that we have access to a number of different editors. The cartoon that I showed earlier talked about Cas9 on the left, and that's kind of version 1.0 of genome editing. There's been version 2.0 and additional engineering of this type of system to do very elaborate types of modifications. So in the middle, there's RNA editors. There's editors that can change single bases very well, called base editors. There's additional ones that are called prime editors. You can change the epigenome, meaning not necessarily the sequence, but also how the genome is organized and packaged. But for simplicity's sake today, I'll focus on Cas9 and just know that this field is rapidly evolving with billions of dollars really being invested in all of these technologies. One of the nice stories and efforts that we have on campus is to try to move away from using viruses to deliver the CRISPR machinery. And so we have some work that's been able to decorate the protein part of it, basically fuse different additional peptides, molecules to the protein, such that we can get desired editing outcomes. And then also, we've been working with Dr. Gong's group next door to package them into nanoparticles. And on the right, this is a kind of, cartoon version of what we think these nanoparticles are doing once they try to enter the cell. In the middle is the nucleus, but we can control how the payload within these nanoparticles gets delivered, such that the CRISPR machinery can go in and change the genome really effectively. One of these particles is what we call a nanocage. You can see a transmission electron microscope image on the top left. They're about 20 nanometers in diameter. And what we do is we've used a common surgical technique in ophthalmology where you can inject fluids into the back of the eye. And this is called subretinal injection. And the reason for this is that there's a layer of cells in the back called the retinal pigmented epithelial cells, RPE, that has been the site of dysfunction for many types of disorders. So that Best disease disorder that I introduced at the beginning, that is actually that protein is expressed specifically in the RPE. And after we inject into, in this case, the 2 mouse eyes, you can take out the eye after euthanizing the mouse, peel away the retina and flatten the RPE, and thus shown here as a floret. And in this case, this mouse has been engineered such that if you have proper editing, you will see a red signal, red fluorescent protein. And so portions of the eye that have been edited are in red, and you can see there's patches here that are very highly edited. And we can modify the surface of the nanocapsule with various ligands and molecules to enhance uptake into this specific part of the eye. And that's shown on the right. To give you an example of how we actually designed the editor, this is that portion of the genome, BEST1, where that misspelling occurred. That T misspelling here on the bottom is called R218C because that's the amino acids that's been changed in the protein. And you can design guide RNAs, which is that orange component in the CRISPR system, to match specifically only the T base in this copy of the genome, and specifically go in and cut that copy of the genome. And so this is the design that a graduate student of mine, together with a postdoc in David Gam's lab, designed and published last year. And the reason we're going after that specific mutation is that it encodes actually a key portion of this protein that's involved in chloride current movement through this channel. And there's different amino acids in this protein that have been shown to be mutated in different patients. They're numbered here on the left. And the concept is pretty simple. If we can go in with CRISPR and you have one healthy copy from one of your parents and a mutant copy from the other parent, you would produce a protein that has a mixture of both monomers or subunits of that protein that make the channel from both of your parents. If you're able to go in with CRISPR and delete or actually destroy the transcript that would lead to the protein, you could make the entire protein from the healthy copy. So these types of dominant disorders are an attractive target for genome editing. And what we're able to do is generate bits of retinal tissue using stem cell technology. Here on the left, we can generate human pluripotent stem cells with technology originally developed actually in this building by Jamie Thompson's lab here at UW to take skin samples and make them into embryonic-like stem cells. From that state, you can make any cell type in the body in theory, and we've controlled these cells to go in and make retinal tissues. And now we can make specifically that pigmented epithelial cell and add our genome editor. And so when we do all of that experiments, I'm fast forwarding probably years worth of work, and look at what happens in the genome and read out what genomic sequences we get at that specific spot in the genome. We certainly see half of the genome approximately be the normal copy, the C base here in the middle. And in the unedited sample, the mutant copy should be the other half, about 50%. But here, after doing genome editing, we've reduced that down to 12% of the genomic copies. And all of the other repair products after cutting the genome end up causing what we call frame shifts that disrupt the protein, specifically the mutant form of it, such that it gets destroyed. Such that really only 5% of the genomic copies can make a protein that would be not the proper sequence. What does this mean in terms of what the cell would do? I mentioned chloride current goes through that channel, and we can look at that at a single cell resolution by taking these cells from not only that R218C patient, but we've corrected it using CRISPR and looked at a number of other patients. And if you do a current and measure the current that goes through that channel, specifically the chloride current, there are these dysfunctional channels that are shown in red. And the normal one, you have a kind of a diagonal line that shows nice current. And our genome edited lines have the proper current as well. And it's actually a pretty complex case. There's various types of mutations that are in different parts of the gene that would be amenable to natural gene augmentation, which would be the older technology that I mentioned at the beginning, where it does not involve gene editing, where you just saturate the cell with additional copies of the gene. But with gene editing, that seems to work for all of the mutations that we've tried so far. And so I wanted to summarize this type of work where we have really robust tools on campus, and we take advantage of the really cutting edge stem cell community that's been developed over decades to now take those human tissues, micro tissues. We have bits of beating hearts in the lab, neural tissues, retinal tissues, muscle tissues that we can add genome editing to and predict before we go into a patient what the consequences of that editing would be. And so this National Center is working together with my lab as well as others in UW to see how this comes together to translate these types of genome editing strategies to the clinic. So thus far, I've talked about the idea of actually injecting CRISPR into the body. You can imagine sometimes that wouldn't go to the right place, and therefore, and you'd like to do multiple rounds of editing, perhaps to add in a lot of functionality. In that case, you could take cells out of the body, manipulate them like Namita just showed you in the video, and then put them back into the body. And in this case, we're focused on T cells. Certainly cells of the blood are easy to take out and put back in. And so one of the really exciting developments in the field has been to generate cellular T cell therapies that have been genetically engineered to go in and find dysfunctional cells in the body. And so what you're seeing here is a T cell attacking its target and is essentially killing it if you had the video long enough. There are 5 approved T cell therapies like this out there at the moment that oncologists look to for treatment. And then there's thousands in development as well for other cancers, as well as solid tumors. Even Alzheimer's and aging has been thought to be a potential indication that can be treated using these T cell therapies. Again, this is a large team effort. We're part of a center from the National Science Foundation called CMAT, or the Center for Cell Manufacturing Technologies, and also a dream team, the Pediatric Cancer Dream Team from a philanthropic organization, Stand Up to Cancer. And you can see we're, again, fortunate to have a really esteemed set of colleagues that help us design and test these types of strategies. On campus, we've looked also to develop new capabilities to not only discover these types of strategies, but also to try to manufacture these here in the hospital. And so this is in partnership with all of these folks on campus, namely the Program for Advanced Cell Therapy here, led by Jacques Galipeau and Ross Myers. All of the work that we're doing here at WID is being transferred, and we're at the process of seeing whether we can replicate that in the hospital in actually a clinical setting, such that these types of products could be entering clinical trials here at UW. So the technology that's been developed by graduate students in my lab, namely Katie Mueller and Nicole Piscopo, in collaboration with Christian Capitini, who's a hematologist oncologist, is to use CRISPR. Again, here on the top, we're cutting the genome, and instead of putting a short single base pair change, we're putting in thousands of bases. And we can do that because we can take T cells outside the body and grow them up and manipulate them and purify out the ones that have been edited. In this case, we confer what we functionality, what we term CAR, or chimeric antigen receptor, that allows these T cells to recognize a specific type of cancer that has a GD2 target molecule. GD2 is turned on in a number of solid tumors, including sarcomas and neuroblastoma, which is some of the cancers that we've used to test this type of T cell product. We want to make sure that we're cutting at the right spot in the genome. And so there's an assay where we can look throughout the genome to see where we have DNA double-strand breaks and count how many times do we cut in every single chromosome. So this might be hard to see, but you have 23 different chromosomes, 22 plus X or Y, that we've mapped where the genome edit occurs. We have very nice specificity here where we intend to cut, which is the T cell alpha chain. And that has the sequence highlighted here on the top. And there's 20 bases in that guide RNA that confer the specificity to that spot. There are other places in the genome where you may not have the exact 20 bases next to one another, but you have maybe 19 of the bases being the same. And so that's a potential for what people call an off-target edit or modification. And we'd like to know whether that happens. And all of the different areas in the genome that have either single changes that are highlighted in the different colors here, or multiple changes, there could be 3 or 4 that are mismatched. Those still have a possibility, even though they're 10 to 100 times or 1,000 times less likely. We can detect that now to make sure that the types of edits that we're doing are really targeted to where we intend them to be. One of the nice things about this type of work is that we can control the type of cell to some sense using CRISPR. In this case, T cells exist in a number of different states. They mature from a stem cell state to a more exhausted state. And in the clinic, patients that have more higher percentage of stem cells, T cells, are the ones that respond better to treatment. And indeed, the cells that we're making that we've termed Ferrari CAR because of an acronym that was shown in the previous slide, these actually have a higher memory phenotype when we look at all of the RNA in these cells. We've matched them to the state of the art that's in the field, which is using viruses. And viruses seem to exhaust these cells more so than our CRISPR technique to put in this receptor. So does this work? To our knowledge, it works quite well in these animal models. So what is shown here is a mouse where we've taken a neuroblastoma cell line taken from a patient and generated that tumor in the mouse. So these are human tumors within a mouse host. And then we add our T cell products such that those T cells can go in and eliminate those cancers. We are the top 3 mice have been treated with our gene edited product, and the arrows here show that these tumors have been eliminated at day 25, also as early as day 18. So if we track the survival of these mice over time, the ones where we've treated it with our CRISPR CAR T cells have the highest probability of event-free survival. We do have some complications when we have both the T cell receptor that has not been knocked out in the viral standard product versus our CRISPR product. And so this is really new work that we're putting together for a manuscript submission really in the coming weeks. So I want to make sure that we have some time for questions. I'll finish here. I'd like to highlight that gene therapies involving CRISPR are already in clinical trials. There's both the form of CRISPR T cells that are being tried in patients, as well as directly injecting CRISPR to treat cardiovascular disease, metabolic disease, infectious diseases like HIV, and a number of inherited disorders. There's one in the eye that's probably the first of its kind by Editas. And in general, I would argue that viral delivery as a modality to put in genetic functionality is here to stay. However, for some applications, some of these virus-free strategies to change the genome can be quite flexible and easy to manufacture. And you certainly saw the case of that in terms of delivering antigens for vaccine development, where they use entirely virus-free methods. I think similar stories could be said and pointed to in gene therapy and cell therapy. All right, and I think I'll finish here by acknowledging really the heroes that have done all of this work. The eye work was really started by Ben Stair, who was an MD PhD student who's now graduated at University of Pennsylvania. And the T-cell work was initially started by Nicole Piscopo, who's a scientist at CRISPR Therapeutics out in Boston, and Katie Mueller, who's a graduate student in molecular biology here.
Great. Well, thank you, Professor Saha. This is really fascinating stuff and really exciting about the progress that's being made. So we have a number of questions. And I think, and you touched on this, but I want to go back because I think people ask this a lot in terms of how, we're already seeing some therapy, you said, happening in the clinic, but how close are we to addressing, the myriad of cancers? You mentioned Alzheimer's. Folks have asked about Parkinson's disease. How close are we to this really being, I can go into my doctor and they can do this work for cures directly?
Yeah, I think we're very close. I mean, there's certainly, I think, staggering investment into this technology, and so that will put a lot of shots on goal in terms of different ways of delivering CRISPR into the body. The programs that are most advanced are in the eye. So there's, I mentioned, an injection into the back of the eye. So that's being in clinical trials, and they reported their results just a few months ago. So that's in phase 1-2. There's a few others like that are in different parts of the body. Heart disease, Verve Therapeutics is one of these companies that's, I think, planning to start in the next few months. And the typical clinical trial, I think, takes on the order of 5 to 7 years. For some of the rare disease orphan scenarios and breakthrough labels, it can be shorter. So that's roughly the timeline for various products to be approved. I do think some of the ex vivo T cell therapies out there, off the shelf cells that doctors can point to treat, say, a cancer, are also promising and in a similar phase, maybe a bit further along. because you have additional opportunities to check that you're editing properly, merely because you have the opportunity to look at the cells outside the lab and before they go into a patient. So there are products out there, as I mentioned, the T cell products, as well as natural killer cell products. There's a really big push to make universal cells from stem cells that would not need to be matched for antigens, such that you don't have graft versus host disease. And those projects are coming along quite quickly. These cells could also be used in kind of devices. So there's, I think, promising treatments for diabetes that were, in the newspapers the last few weeks ago. And you can imagine that all of those cells could also be subjected and designed in some ways using the genetic engineering and CRISPR technologies that I mentioned. So, the short answer is, if it's just CRISPR as a therapeutic, it's probably a few years, probably 5 to 10 years is the most realistic estimate. But CRISPR is a tool that's being already implemented in all sorts of drug discovery programs, and probably will be intersecting with tissue and tissue engineered therapies and medical devices as well.
Great. So I'm clear. You talk about what you sort of mentioned off the shelf, but there's ability to produce cells from the pluripotent stem cells that they're in a product that's injectable in some way, but also there is this sort of customized ability to take an individual cell and manage that. Could you maybe just sort of explain those differences so I'm clear, and I think others are clear on those?
Yeah, that's a very important distinction and an interesting one as well in terms of strategy for different companies out there. What I mentioned in the second story was T cells, and those are patient matched. You can take out the cells from a single patient, send them, FedEx them to a central manufacturing facility. They will put in the receptor, and that exact batch of cells from that patient will go back into the same patient. That would be called autologous, and that's very expensive, as you can imagine, because you have to generate a custom product for every patient. That said, there are 5 products out there where that happens, and many in the industry think that's is here to stay, at least for the next decade or so. The cheaper, arguably cheaper way to do this, and simpler way to do this, is to have a single batch of cells that could be used for multiple patients. And their embryonic stem cells are attractive because they can grow indefinitely in the lab. They've already been grown for nearly 20 to 30 years, and in theory they can grow forever. So if we're able to make therapeutic cells from that type of cell source, then doctors would have a frozen set of cells, sitting around on the shelf in the stockroom, and they'll say, okay, let me prescribe a cell dose. And so that is a vision for some of these companies out there. Fate Therapeutics, for instance, has a natural killer cell product that's in trials that has a similar type of recognition of cancer, but it is not autologous. It is off the shelf. And it might be one day where you have multiple types of cell therapies, fighting the same indication.
Great. Yeah, that's great. So I imagine being here in Madison, again, where stem cell work was pioneered, and those connections really helps in your research and your work, as well as having the Carbon Cancer Center and other connections across campus, must make that collaboration a lot easier for you in your lab.
Oh yes, definitely. I think the support that we get from the foundation to try some of these new ideas is really essential, because as you can see, the design of this really does involve deep conversations with clinicians, and it needs to be tailored in many ways to the exact patient ultimately, but also to the exact tissue type of biology that is behind it. A single amino acid in one place of the protein actually had a big effect on how we design our therapeutic. And so those types of conversations are really enabled by some of the cross-campus programs and centers and institutes that I named here at the last slide. It's been really a real pleasure to see the campus come together around this type of technology, and to see places where we can kind of have the Wisconsin idea be put into action, because ultimately we're not doing this work just to keep it in the lab. We want to really grow it here in Wisconsin, move it towards clinical trials on campus, and that's why we've been put in really all of that work with the partners at the hospital to see whether this technology can help the people in the state as well as around the world.
Great. So I guess that leads to a question that I think others have asked about, was sort of the ethical component of this. And this gene editing could lead maybe to some ethical challenges. So I wonder if you could sort of talk about, your work in that and sort of the guide rails that exist around making sure that we're doing this work for the positive benefit, and we're not sort of crossing maybe some of those ethical boundaries.
Yeah, I mean, it's a very important question, and a difficult one, actually, that the field, I would say, is still debating. I do have a slide or two. Let me move to it. We talked to many folks on campus, including folks here in the Science and Technology Studies Center, the Holt Center, and the Department of Medical History and Bioethics. And from the very first days of CRISPR, when I started my lab in 2012, this idea that you can not only just inject CRISPR into the body as a therapeutic, but rather use it in reproductive medicine to change every cell in the person, has been on the horizon. And so this image here, what you see is the major distinction in the field, which is somatic editing, which is in kind of the green column here, versus germline editing, which is in the red column. And the germline editing is applying CRISPR to embryos that then would be transferred to a woman's womb to give birth to a child that's been entirely edited. And that has been the subject of international summits. There have been calls for moratoria on that. And importantly, my lab does not do any of the germline editing experiments, and federal funding for that type of experimentation involving the potential destruction of embryos is also forbidden. That said, there have been pretty notable experimentation that's still gone forward in that column. There's one that made international headlines in 2018 that came out of China. And so there's a question right now is, what are the guardrails to doing this? And even for somatic cell editing, how do we ensure that we're doing it really only for therapeutic purposes, not necessarily for enhancing particular traits? For instance, if we were to confer different types of vision in addition to 20/20, see infrared has been proposed. Is that really therapeutic, or is that enhancement? And should our field really be working towards those applications? So I've put a few questions there that can prompt some I think, reflection from folks in the audience. And we're also part of a effort that's around the country that's centered at Harvard, but also Arizona State University, called the Global Observatory for Genome Editing, that we're going to have events and workshops. We actually have one tomorrow, and this website is being launched next week to start to expand the conversation about some of these questions. So, I would say it's still an important kind of question to reflect on, not only by scientists, but by many others on campus.
Great. So, and let me go to a question that one of our audience participants asked, and hopefully I can do a good job of explaining it, but I think it sort of relates maybe to the question. So the question is, procedurally, do the edited cells that you introduce into the body or specific organ seek out and modify a large percentage of all disease cells with this typographical error in their structure? So I'm guessing the question is around what is it very targeted in that finding a solution, or can it fix a broader array of disease cells? I think that's what the question gets to.
Yeah. So I mean, it does depend on the disease, and there are strategies to do either type of scenario. The strategy I discussed in the eye is quite targeted. It's a small portion of the retina. However, that could have a large functional effect on the patient. They might be able to have much better quality of life if they can see incrementally better. There are diseases like muscular dystrophy in particular, where you actually need to hit most of the body. You can't just hit a small portion of the muscle and expect the patient to recover. And that's where you would have a systemic infusion of CRISPR to try to hit every single muscle cell in the body. You can see that's a tall order. However, I think there's progress there, and it turns out that the doctors think that you don't need to hit every single cell. Even having like 5% of the muscle being gene edited could have a therapeutic effect. And so there's clinical trials, I believe, underway, certainly in gene therapy, and there are efforts very close to clinical trials using gene editing for muscular dystrophy.
So the theory behind that is if you improve some of the muscle, then that helps spread across, or the theory is that you only need to improve a certain percentage of the muscle to regain maybe full function?
Yeah, it's probably more the latter. However, one of the very interesting concepts out there is to try to target stem cells that end up making a lot of the muscle. So if you're able to hit, in particular, stem cells in the muscle called satellite cells at high percentage, over time that can contribute to more and more muscle. And eventually, all of the muscle that's generated from your stem cells would be gene edited. And so there are specific structures in the body that we're really trying to deliver to. Hematopoietic stem cells, for instance, is a large target. If you're able to hit that, within 3 weeks, your entire blood system could be gene edited. It is a tall order. Those cells sit in the middle of a bone marrow, surrounded by a bunch of other cells. It's hard to deliver, arguably, this type of machinery into that specific minority of cells.
Okay, great. So another question, and maybe a little bit related, I know you talked a little bit about sort of using this to treat tumors. So if someone's got like a brain tumor or other tumors, your ability to essentially go in and destroy that tumor. And I don't know if you can talk a little bit. I know you mentioned a little bit around that work.
Yeah. So the adding a receptor, the tumor has surface characteristics that biologists can find that are different from healthy tissue. And as soon as you know those characteristics, you can engineer these types of receptors into a T cell. And the T cells, once they're in your bloodstream, actually can cross the blood-brain barrier and get into the brain and surveil portions of the brain and find that characteristic on the surface of the tissue and trigger a cytotoxic response, because T cells have these natural abilities to kill target cells. That's what they're trained to do. And we're just simply rewiring that using our gene editing. This has been, I think, game-changing for leukemias, and the hope is that we could do this for other cancers, including brain cancer. And we have a program on this focused on glioblastoma.
Okay, great. So I wonder a question about, and you mentioned a little bit about the mRNA and a lot of the work that was done during COVID to come up with these vaccinations that we have with COVID. What sort of work in that area is sort of helping drive some of the gene editing? Are there learnings that we can take from sort of that great work that's being applied to the work you're doing?
Well, I think some of the delivery strategies that have been used in the COVID vaccines, namely from BioNTech and Moderna, could be repackaged to put in genome editing components. That's one potential place that it could impact gene therapy. Delivering to the right cells, as we just discussed, is a big challenge in the field. And so, some of these carriers that have now been tested in, arguably hundreds of millions of people, could have be reused for gene editing. I think also the manufacturing supply chain struggles, as well as opportunities where they were able to innovate, also could be useful for how we can deploy gene editing at a larger scale.
Great. So maybe one last question, and you talk about a lot of what, I guess is the exciting part about this work is that it has an ability to impact a lot of health, a lot of areas that have sort of challenged the medical community and others for a long time. From where you sit in your lab and the work you're doing, what's the area that excites you the most? What's the area that you think really is the piece that's going to be the most dramatic or the most impactful in the near future?
Yeah, I mean, I think the two stories are the ones that I'm super excited about. I do think that the rewiring of the immune system has a lot of promise, not only for cancer, but for other kind of dysfunctional tissues or components of the body. So we are working on engineering T-cell therapies to go in and sense aged tissue, scar tissue, tissue that's been maybe riddled with plaques in Alzheimer's brains with some engineered immune cell therapies. So delivery to the brain in particular has been a large challenge for any type of therapeutic, small molecules included. Some of these cells have the ability to get through the blood-brain barrier, get into the brain, and deliver and secrete factors that could be therapeutic. So it's kind of a Trojan horse to bring in new types of functionality into tissues that really need, I think, a lot of intervention. The nice, also surprising thing about the T-cell therapies in the patients is that it can be durable. Years later after infusion, those T-cells have these memory stem cell characteristics and stick around in the patients. You can detect them. And so these are, arguably durable curative type of strategies that are truly exciting to develop.
Right. Great. Well, I think we have come to our hour, and Professor Saha, thank you. I know you've got a very busy schedule, so we really appreciate you taking the time to spend with us and explain this. Again, really exciting work that's happening, and the work that you and your team in the lab are doing. And so we really appreciate your time and your effort with us today.
Yeah, I appreciate the opportunity and the spotlight. Thank you.
Great. So I will say to our audience again, we have recorded this video and we will be posting it to YouTube. We'll send you all, if you registered, a link. So you feel free to go back. I know at times some of the science in the slides can take a little bit to understand. So you'll have an opportunity to go back and look at that again, and feel free to let us know your questions and concerns. So with that, I will thank you for joining us today and being part of the Wisconsin Foundation Alumni Association Wisconsin Ideas Series. And we wish you all a great holiday season and a Happy New Year.